So is the screen visible?
Yes, ma'am.
Okay. So today's class is on mammalian host cells and expression vectors, and I hope you understand the importance of mammalian vectors as well as the host for making copies of the recombinant protein that you might be working on. Because you have been repeatedly being told that in E. coli, the post-translational modifications are not there, the ones that we find in humans. So then you were also introduced to the fact that not always E. coli is used for recombinant protein generation, but if you're having problems with the post-translational modifications of certain proteins, then you can go for yeast expression systems because it is a eukaryotic expression system. So in that context, I had introduced you to Saccharomyces cerevisiae, and then I told you that even in S. cerevisiae there is some problem. So then I told you about Pichia pastoris, and I told that how the post-translational modifications of the recombinant proteins in Pichia is very much similar to what we see in humans. But is it exactly similar? Is it the best strain that we have? No. I also sensitized you to the fact that people are still looking for alternatives for these various expression systems. In that context, there are a few other yeast strains that are being pursued uh, in research for seeing whether they can be very good host strains. Then there are some fungal species, etc. So means people are still trying to find the best alternatives for recombinant protein production. And in fact, means today's class is on mammalian cell lines, which can be used for recombinant DNA production, protein production. So first, means I know that you have been told about this many times, but again, I also want to tell to you one fact that it's not always that the problems are associated only with the, let's say, post-translational modifications with respect to protein expression in E. coli, but it could also be the sequence of the foreign gene that can be a problem. So means let's just go through again with the various problems that we face when we produce a particular recombinant protein in E. coli. So there can be certain problems with respect to the sequence of the foreign gene. So one is, of course, that because in E. coli, it being prokaryotic, there is no splicing. So if you clone a genomic sequence, considering that, okay, if I just clone the genomic sequence of my gene that I'm interested in E. coli, then obviously it will not be processed further and we will get incorrect or truncated polypeptide. So in those cases, do we have an option? Certainly, we do have an option that you clone the cDNA rather than the genomic sequence. But yes, this can be considered one problem because then you are totally dependent on first generating the cDNA. So your cDNA generation has to be very efficient, it has to be correct, and only then it can be introduced into E. coli. Then second problem that is seen when people have tried to overexpress these foreign gene in E. coli is that because of the various different regulatory features or sequence features that differ between eukaryotes and prokaryotes. So there is a possibility that there is some region in the foreign gene that can be recognized as a terminator sequence in E. coli. And what do we would see in that scenario would be a truncated transcript as well as the protein. So yes, I mean this problem is also seen in many cases, and how this slide has been generated by various experiments that were done by various scientists in different labs, and it is their individual experiences that finally we can say that yes, so these type of problems are also seen when you express your protein in E. coli. Then it is also seen that many times that because of certain sequences of the foreign gene which are present in the N-terminal domain, the protein production is not apt. It is not good. So in those cases, it was seen that the sequences in the N-terminal were making it, let's say, more unstable in nature, or because of the sequences which were present on the N-terminal end, the translation was not very efficient. So, do we have an option to correct this problem? Yes. In that context, I will introduce you to the fusion peptides that are generally used, means signal peptides, etc. So, next week's class will be on that. But at the same time, you have been introduced due to the fact that you can make fusion proteins in which either at the N terminal end or at the C terminal end, you can add a tag, and that tag may serve various purposes. One of the purpose that was told to you before and also as part of your assignment was the fact that you can add GFP on either side of the protein, and because of GFP you can monitor your protein under microscope, and you can check its movement. You can see where exactly that protein is being expressed. So that is just one of the advantage of the signal peptides or tags that are used. But coming back to the problems faced on the for the foreign protein production, it was seen that by addition of certain fusion tags, the stability was dramatically better. And similarly, the translation was improved manyfold when certain signal peptides belonging to the prokaryotic origin were added on the N terminal end. So this is one problem that we face, but how can be corrected? I told you can add the tags, but what about these premature termination signals? So these signals also, they can be corrected by the way of site-directed mutagenesis. So your next practical or assignment lecture will be on site-directed. Sorry, it will be on siRNA and shRNA production, and after that we'll have the site-directed mutagenesis. So in that you have to do it smartly, that you should not make such mutations here that it leads to the production of stop codon, but you have to means obviously take the sequence into account, that if you are making the change, is it means there can be certain constraints. In some cases, probably the mutation that you'll have to make, probably it will change amino acid. And if it changes the amino acid, maybe that particular amino acid is very, very important for protein function, or it is extremely important for protein stability. So in those cases, you'll again have to think about what sort of mutations you have to do and all. But yes, you do have the option to correct the this premature termination signals in the foreign gene. The third problem that is generally seen when people express the foreign gene in a different host. It means in this case, let's say a mammalian gene in E. coli, but this problem of codon bias is not limited to that. Means it is that any foreign host that you want to express your gene in, there can be a problem of codon bias. So what is codon bias is simply the fact that certain organisms have certain inclinations towards certain these codons which code for a particular amino acid. For example, in human genes, it was seen that generally CCA, CCT, or triple C, these are the ones which code for proline. Means in the human genome, in the coding region, you will generally find these codons which will code for proline, right? But in E. coli, it was seen that it is majorly this enriched in the codons CCG. So CCG again codes for proline, but in E. coli, for whatever reason, rather than these other codons which code for proline, you find a more prevalence of double CG. So, what is the implication of this? Would be the fact that the tRNAs which recognize CCG to get the proline opposite to this codon, they are more prevalent. While the tRNAs which recognize CCA, CCT, or triple C, they are comparatively much less prevalently present in E. coli. So, what will happen that if I clone this human gene in E. coli and I expect that the protein expression will be very good, I may not always see that because it will be limited by the presence of tRNAs for that particular codon. Because they are not sufficiently present, there might be a condition that there is some lag, or it is slow protein production, or it is not good to that extent. So in those cases, can we correct this problem? Yes, again, through site-directed mutagenesis, what people do is that they try to change the codons which are present in the foreign gene. For example, if it is CCT here, through site-directed, they will change it to CCG, and then this problem will no more be seen. Alternatively, what we can also do is that we can engineer our host strains, for example, E. coli host strains, in such a way that they start expressing the tRNAs which recognize these particular codons, the human codons. So both strategies have been used, and there are strains available of E. coli which have these engineered tRNA molecules in those. Now, other problems that are faced means many times it will happen that, let's say, you have used your proper cDNAs there. The sequence is completely correct. There is no premature this termination thing. There are no sequences at the end, terminal end, which will make it unstable. At the same time, let's say in your foreign gene, there is no problem of codon bias either. But still, you will find that your protein is not being present at high levels, or let's say even if it is expressed at very high levels, then it is inactive. So then comes these problems which are caused by E. coli, such as that the recombinant protein which is made, maybe it is not being folded properly, and there are incorrect disulfide or not sufficient disulfide bonds being formed. And there is a possibility, see, many times what happens that the protein, the final protein that we want to use, let's say for therapeutic purpose, it needs proteolytic cleavage. So there are precursor polypeptides that are made from the gene, and from the precursor polypeptides you get the mature protein, which is finally used for whatever purposes you are interested in. So there is a possibility that E. coli in this cleavage is not taking place properly. Then obviously there are so many these post-translational modifications, glycosylation, phosphorylation, acetylation, and so on, which are very, very crucial, maybe in some cases for their stability as well, and also for their functioning. So if these things are not there, then having an inactive protein is no good. So in those cases, definitely you'll have to go for mammalian systems or eukaryotic systems. So in this case also, yeah, one option is that you go to mammalian system, or in E. coli system only you make changes so that these problems are not much seen. So you will still find many reviews in which you will see that E. coli strains are being engineered for them to, have better post-translational modifications. Or let's say if your protein is getting degraded, then they are being engineered in such a way that they are protease deficient strains. Or let's say if your protein is not getting folded properly, then in the host strains, what they are doing is they are overexpressing chaperones, which will ensure that the protein of your interest definitely gets folded properly. So, yeah, means again, I will emphasize that it's a dynamic field. E. coli proteins as source of human origin recombinant proteins is not a closed chapter. Many people are still trying to do it. Now, coming to eukaryotic system, I've already told you about the yeast strains, etc. Now, coming to the mammalian systems. So, in mammalian systems, which are the systems that we generally use, so there are various cell lines which are available, which are used for recombinant protein production. So, what is the source of these cell lines? Are they only humans? No, in many cases they can be from different mammal as well. These are the general, very, very commonly used cell lines. So I've given some details of those. So Vero cell line is derived from African green monkey from the kidney. So you will see that many of these, they are from the origin is kidney. So these kidney cell lines, Vero is very much used for even this virus infection of these cell lines and recombinant protein production, etc. Similarly, this is baby hamster kidney cell line BHK21, which is also very commonly used for expression of biopharmaceuticals and also for virus propagation, etc. And one which is most used, and you will find in almost all the mammalian or cancer cell biology labs, would be the HEC293 cell lines. And I'll just discuss briefly about this. And these are the cell lines which are generally used for transient expression of your gene of interest. And CHO cell lines are the cell lines which are the most used for long term or stable expression of your gene of interest for therapeutic purposes. Means all the mammalian cell bioreactor labs would definitely have CHO cell lines. So I just detail you about the HEC293 and CHO cell lines. But yes, still people are trying to find more cell lines which can be better than the same. But I would still say that as compared to the unicellular organisms, be it yeast or bacteria, where people are looking for alternative organisms for overexpression of recombinant proteins. In case of mammalian cell lines, it is that people are making changes in these cell lines to make them better. Means somehow the discovery has gone in this way that there are not many papers where they have looked for better cell lines than the ones that have been already created, but rather than that they have made changes in the cell lines itself to make them better host strains. So these are for your notes. Please go through these. So HEC293 are human embryonic kidney, means they are derived from the embryo, at the embryo stage, the kidney tissue, the kidney cell lines. And then why 293? Because it was the 293rd experiment of the scientist who was working on it and who derived these cell lines. So under microscope, they look like this, and they can grow both in as adherent cell lines, means you can plate them in a tissue culture plate and they will expand when given a proper media. And also some changes have been made in these cell lines that they can also grow as suspension cell lines, that if you have multi-liter these bioreactors to grow them in suspension also, you can grow them. And why you need to grow them in very large bioreactors and generally would prefer not them not to be adherent is simply due to the fact that much more cells can be grown in suspension as compared to when you are limited by the area of the tissue culture plate where these cells are growing. So one point is that so these HEC293 cell lines, they have been used for producing very high amounts of recombinant proteins. And these cells, when they were derived, they were transfected with the shared adenovirus DNA. And it is then that these kidney cells, which were non-tumorigenic, they become transformed in nature, or let's say they became immortal in nature, so that they can be repeatedly grown in the lab, and they will they will not die. So in that context, that's why it is called as a cell line. But otherwise, if you derive any cell from humans, they will have a finite life. After some time, they will senesce and then they will die. So, but in case of the cell lines from mammals, any of the mammals, be it humans or hamster, etc., uh, they are called cell lines because they have been immortalized. In some cases, it was found that it was some random mutation that took place, but in many cases, they have been immortalized by adding certain proteins from viruses, which interfered with the cell cycle of these cell lines, and also the apoptotic, which is a programmed cell death pathway by which they became immortal in nature. So you can read the rest of the notes for you, that what are the specific advantages of these cell lines, why specifically HEC293 cell lines, or let's say not other cell lines, for example, the ones that I'm working in the lab and all. Why not all human cell lines are equally good? Because obviously, like in case of other organisms also, there are certain organisms which are comparatively more hardy. They don't need that much of extra care or extra nutrients for their growth as compared to HEC293, sorry, some other cells. So that's why 293 cells they are used. They are pretty, they divide comparatively rapidly, doubling every 36 hours. You can imagine what rapidly means in context of mammalian as opposed to bacteria. So in bacteria, you know that if given proper conditions, E. coli divides every 20 minutes. But in case of human cell lines, they divide every 24 hours or 30 hours, depending on which cell line we are using. So that's why bacteria would be the first choice. If your protein of interest is getting, this amply produced in bacterial origin host strain, then it is the best, because growth of mammalian cells, it requires serum, it requires tissue culture plates which are specially made and all. So growing a mammalian cell culture versus a bacterial cell culture is requires lots of money. So that's why bacteria or yeast cultures are preferred as compared to mammalian cell cultures. But in many cases, because your final aim is, let's say, for therapy, if the protein is inactive in bacteria and yeast, then obviously there's no point in going for those. Then you have no option but to go for mammalian cell culture. Okay, then then I told you that these cell lines, there are many variations to it. Another one which is very commonly seen is 293T. So in this 293T cell line, they have introduced viral origin large T antigen, in which the plasmid vectors, which have SV40 origin of replication, they replicate very fine, and you can get large amount of proteins. And also it was seen that HEC293 T cells, they divide much faster as compared to HEC293. So these are the cell lines which are used for recombinant DNA production. Another cell line which is most commonly used for recombinant DNA production is CHO cell lines, mainly for stable cultures as well. So, as the name suggests, so it is Chinese hamster ovary cell lines. The details are given in the notes section. So, these cell lines, they were altered. This is the most simplified diagram I could find. But if you find that how many variations of CHO cell lines have been used, it is a more complex diagram and many more variations have been introduced. So, the one very commonly used is DG44, And at the same time, another cell line was made, DUXB11, and but DG44 is preferred. So what are these cell lines and how do they differ from the original ones? So in the original ones, what they did was that they performed mutagenesis, and these mutagenesis were based, they were differently done. So in this case, chemical mutagenesis was done, in this gamma radiation was done, and the idea was to this mutate a gene called DHFR. So DHFR gene is the one which, uh, if the cell line is deficient in DHFR, then it is totally dependent on growth in a media which is supplemented with glycine, hypoxanthine, and thymidine. So this way they introduced a selection system. So the CHO cell lines in which you introduce your plasmid, which has a DHFR gene, only those CHO cell lines will be able to grow in a minimal media, while the original CHO cell lines, which do not have, which have not been transfected with the this a vector, plasmid vector with DHFR gene, they will die in minimal media. Okay, so this is a selection system that was introduced. And why DG44 was preferred as compared to this? Because in this, both the alleles of DHFR, they were removed. They were mutated actually.
And ma'am,
And
one
this
more
is
doubt.
the suspension. This is the suspension cell line which was made for the CHO cells. Now, coming to the vectors. So vectors, there was one class which Rahul had introduced you to as part of your assignment section, or practical, I would say, your introduction to this particular plasmid pcDNA 3.1. And as you can see in these plasmid vectors, mammalian expression vectors, what all things would be there. There will be a promoter, obviously, which will be recognized by mammalian RNA polymerases. Is the promoter always PCMB or SV40? No, in many cases. So I would say that when we talk about promoters, many general or commercially available eukaryotic expression vectors, they indeed have these promoters from viral origin. But there are many promoters which are also derived from human origin, so which are very highly expressed. So the list of those promoters is given in this link. Okay, so you can go through this link just to have an idea that which promoters are commonly found in eukaryotic expression vectors. So CMV and SV40 you will see very commonly, but in other vectors you will also see certain human origin these promoters as well. And which promoters would these be? The ones which are highly expressed, or which have a very high expression. So are we limited to only those promoters which have very high expression in the expression vectors? No. If you want to go for some low expression vectors also, we do have these promoters which don't express to that high amount. Why would we like to go for those vectors which don't express in that high amount? Maybe in those cases where you see that the protein may have some toxic effects, etc. And also in some cases, you don't want to overwhelm the system. So people go for both the approaches and find out that which system is better for them for production of the protein. Okay, and the origin of replication, we have generally SV40 origin of replication is there in various eukaryotic expression vectors. Then, because many of these are shuttle vectors, that is, that they can also be grown in bacteria as well as in eukaryotes, they will also have origin of replication from plasmid. Now, apart from origin of replication, which is always present in the any cloning or expression vector, obviously, if you want expression vector, you would require a promoter, but at the same time, you want that your gene of interest, when it is getting expressed, means when it is getting transcribed by a promoter, there should also be a transcription termination signal. So which are those termination signal? Mostly these are polyadenylation signal. And I guess I've told you before that there are certain these polyadenylation signals which have been derived from various genes after a lot of molecular biology studies, and it was realized that okay, these polyate signals, like in this case from bovine growth hormone gene, they are very, very good polyadenylation signal, means very strong in nature, which will ensure that whatever gene you have cloned in your expression vector, it does terminate at a particular sequence. Why do you need a termination sequence? Is also because of the fact, because generally what you clone is the coding sequence, which is from the translation site, start site, to translation end site, that is stop codon. So there is absence of the transcription signal, termination signal in your gene of interest. So that's why you need a poly-A signal. In this, you only see one poly-A signal, but in many cases, you will also find two poly-A signals to make it that, okay, if this doesn't work, then surely it will work in the, at least the other one will surely work types. Then there are multiple selectable markers as always, and regarding the fact that you want your transcript to also be translated, because in many cases your final product will be the protein. I'm saying that in majority of the cases it will be protein, but obviously people who are working on non-coding RNAs, etc., your final product would not be protein, and in those cases you do not worry whether you have a Kozak sequence or not. But if your final product has to be a protein, you have to ensure that there is ribosomal binding site, etc., and the details for those have been taken care of in the lectures from the assignment or the practical, so you can please go through it. Apart from the fact that it has selectable marker for eukaryotic cell, they also have selectable marker for bacteria. Just keep this in mind, because it's a shuttle vector. You need its selection as well for bacterial system. So this is for your information, the various selection markers that are used for mammalian expression vectors. You can go through it. Generally, you will see that these markers are against or let's say these resistance genes. These resistant genes, they are against a selection agent which either damages DNA or protein and thus leads to death of the cell. So unless your mammalian cell has taken up a plasmid which has a resistance gene against it, it will not survive. So that's why these are selectable markers. So this is for your information. You can please go through it. Now the thing is that, okay, you have your gene of interest which you have cloned in your expression vector, but then how do you introduce into your eukaryotic cell? So in one of the assignment lecture, you were already told about that you can use liposomal based introduction of your gene of interest or plasmid inside the mammalian cell, or any mammalian cell, need not be human, right? So just want to introduce you to the fact that which are the various transfection methods. Is it only limited to liposomes, or do we have other methods like we had in plants, we had in bacteria? In bacteria, we had two options. You can do transformation. You can introduce your gene through bacteriophages. You can also use electroporation, right? Similarly, for yeast, you have two options. For plants also, I told that there are options in which one case you can use various physical methods. You can use that biolistic method, DNA coated microprojectiles, or you can use chemical methods in which you complex your DNA with these polyethylene amine or some cationic agent, and then you incubate it with the plant protoplast, and then the DNA will be taken up. Or you can use agrobacterium mediated transfer, etc. So there are multiple methods for various organisms, and similarly it stands true for eukaryotic cells also. So the most commonly used method for transfection currently is liposomal based. So you generally now what we do is we buy the lipofectamine from commercial these sources, and you that details you have already been told. You incubate your DNA with liposome. Sorry, this lipofectamine. Then micelles are made. Each micelle will have this DNA incorporated into it. Then this will fuse with the this plasma membrane. It will be then entered inside, and it also goes to the nucleus that has already been checked. So whether so you whenever let's say you are making a new transfection reagent, you also have to see whether your delivery agent is able to take it only to the cytoplasm, or it can take it to the nucleus also. So if it is means obviously if you want the protein to be expressed, then obviously you need it to be going to the nucleus. Only then it will be transcribed and translated, right? Okay, so this is the most commonly used method, and the method which was previously used before, let's say, liposome era was calcium phosphate based transfection. Then, if we have a very good system for liposomal based entry, then do we really need to go for the other methods? Yes, maybe in some cases people try other methods as well. And especially for those methods in which liposomal based transfection is not very good. Like in many primary cells, it's seen that liposomal based method doesn't work very well. It is only for the transformed cell lines or cancer cell lines riposome based entry of DNA works very well. So in those cases, you definitely go for virus-based infection. So do all type of viruses work equally? No, I just come to this point. But then let me just tell you. So one is calcium phosphate-based transfection. Then it's liposome. Ballistic method-based microprojectile was previously used. Now, I don't see many papers coming out of this, not that commonly used, or electroporation either. But you will find some studies, or maybe in some labs where these techniques have been, let's say, very well standardized for one type of cell line that they're working on, then they just continue with the same. Then there is direct injection into nucleus, which is done especially for those when you have to make mammalian cloning. I mean, when Dolly was made, or various these organisms were made, and you had to replace the nuclear material. So in those cases, microinjection is used. So I'll just briefly describe this when I tell you about mammalian cloning. But yes, rest of the things, let me just tell you. So one option is like in case of bacteria or in plants where you do have an option of based infection. If you remember in plant cells, uh, the there was option of using virus based transfer. There was this Gemini virus, and there was Colimovirus. These two viruses are there, which do have the ability to transfer DNA into the plant cell. So similarly, but though it was not found to be that efficient, but in case of human cells, definitely many of these were found to be extremely efficient. So I just come to this particular slide. The details for each of these I found in one of the website, and I've just copied it here for your as part of your notes. Okay. So the most commonly used ones are retroviral, lentiviral, adenoviral, and associated viruses. So these are different viruses. Some are RNA viruses, some are DNA viruses. Okay, one difference is this. Second is they may vary depending on that what DNA insert size they can accommodate. So for AAV, it's comparatively less as compared to the rest of the things. For HSV, if you want to clone very big size, then probably you'll go for HSV, but otherwise it's not that commonly used. And even vaccinia virus is used, and actually the list can be much more, but I'm just telling you about the most commonly used one. Then you can also see that the transfection efficiency, it varies. For example, this virus makes the titer is much high when it infects a mammalian cell, and for this it's comparatively less. But otherwise, still means I would say they are all are very efficient. But yes, there is some difference in the titer efficiency. Then you will also notice that the cell types. You will see that some cell types, for example, retrovirus, it only infects dividing cells. The cells which are not dividing. For example, see, when we are talking in terms of recombinant DNA production and protein production in, let's say, these suspension cell lines. So these suspension cell lines, of course, they are dividing very well. And retrovirus based means delivery of the gene or your construct in these cells would be no problem. But for when you want to do gene therapy, when you want to introduce your gene of interest inside the human cells within the body, means you want to, let's say, inject it in bloodstream, and you want that construct should go to your target gene and then deliver it, then of course there are many human cells which are not dividing at that stage. So in those cases, you will not use retroviruses. And also in those cases, when you want to introduce your gene of interest in primary cells rarely divide. They don't divide as much. So in those cases also retrovirus is not very efficient, etc. So in those cases, you will go for the other ones. And what retrovirus does is that it is a RNA virus. So you clone your gene in the genome of that. And then so means there are certain retrovirus vectors available in which you clone your gene. And then that gets introduced into the retrovirus by packaging, and then it gets inside the mammalian cells. Once it gets inside the mammalian cells, it makes more of its copies. And then it was seen that it introduces the gene of interest inside the chromosomes. But where does it insert? Just like if you remember agrobacterium mediated gene transfer, we don't know. So in this case also, if I'm doing a retrovirus based delivery of gene and I want to generate a stable cell line, I can definitely do that. But only problem would be that where it gets integrated, I don't know. There is a possibility that your gene of interest or your vector gets incorporated, let's say, in a heterochromatic region, which is very tightly coiled. And we know that because there is so much of DNA methylation there, the target gene expression will be extremely low. Even though your this gene sequence is perfect, your everything is perfect there, but what you will find that protein is not getting expressed. Why? Because your stable cell line has been generated in which your gene got incorporated in the heterochromatic region. So in this case, what you have to do is just simply you screen the various clones that have been generated. There is a very high possibility that your gene of interest probably gets incorporated in some euchromatic region. So in that region, it will be highly expressed. But then again, you also have to ensure that it does not disrupt the function of the gene which is already present there. There is a possibility that it interferes with the expression of some gene, important gene for the human cells. So in those cases also, it is better that if you're making any assumption that, let's say, the overexpression of my gene is leading to, let's say, increased cell proliferation. Let's say that you introduce your gene through retrovirus, you make stable cell line, and what you find that your cells have started growing very fast. So what inference you make that because my gene was overexpressed, the cell proliferation was fast, so likely my gene is involved in promotion of cell proliferation. But then you have to be cautious to the fact that this could also be due to the fact that your gene landed in a place of, let's say, some inhibitor of cell cycle. So because the gene which got disrupted was the inhibitor of cell cycle, so as a result your cell proliferation was high. So you cannot attribute it to the function of the gene that you had overexpressed, but indirectly it was through the disruption of the gene that got disrupted. So in those cases, I would just simply say that you go for means evaluating different clones that have been generated. If all of those are giving similar phenotype, which is promotion of cell proliferation, then you can be very sure that the final phenotype is because of your gene. Because it cannot be a random thing that every time it lands at the same place. Okay. So one point is this, that for retrovirus, and this is retrovirus based. Lentivirus is a type of retrovirus. Okay. Now, what is the difference between the two is that for retrovirus infection, it requires a disruption of the nuclear membrane, which takes place during the cell cycle. So because for its infection, it is dependent on not breaking, I mean disruption of nuclear membrane. That's why it can only infect the dividing cells. Okay, so that's why it is better suited for dividing cells for transfer of genes. But in case of lentivirus, which is also a retrovirus, but its entry into the nucleus is not dependent on nuclear membrane disruption. Rather, it has its own machinery of some protein which enables its infection and delivery into the nucleus of the cells, target cells. So that's why it can infect both dividing as well as non-dividing cells. And here also, it's a retrovirus, so RNA will be transferred, and RNA will then get converted to cDNA, and cDNA will get incorporated at a random site in the genome. So it's the only problem in these two cases would be that you have to screen the stable cell lines to see that which is giving us a very good protein expression, target gene expression, and if it is not disrupting any normal gene, or because of its insertion, let's say no gene has become cancerous in nature and so on. So otherwise, these are very good vectors. Then coming to adenovirus. So I'm shortly done with my class. So adenovirus is a DNA virus, and it is very efficient in the delivery of your gene of interest or plasmid vector into the target cells. It is equally good for dividing and non-dividing cells. And but the only problem is that its DNA does not get incorporated into the genome. So it means that if you are delivering any gene through adenovirus, then you have to do repeated transfections, because maximum within, let's say, within a week for sure, you will lose your gene, your plasmid, because obviously the cells are dividing, and with each division, the number of copies that had gone into the plasmid, they'll be halved and then they'll be gone. So that's why it is suited only for transient transfection. And one problem seen with adenovirus is that if you want to use it for, let's say, gene therapy purposes, then it was found to be comparatively immunogenic. Then there is adeno-associated virus. The only point that I would like to stress upon Actually, 2 points. One, that it is very good with respect to the fact that it gives stable expression, because delivery of your gene through this virus also leads to this permanent expression, because your gene gets incorporated in the genome. The good part is that in this case, at least, we know that at what location your gene is getting expressed, because it has some tropism. It only goes to a particular location in the genome and only integrates your gene of interest there. Okay, so this is the property of this virus. So in this case, we are not worried about the random insertion. We very well know where it gets integrated. But the only problem with AAV is the fact that this virus cannot infect by itself, means it can not be packaged by itself. For infection, yes, it can do by itself, but for its further packaging, it requires helper virus. So you have to do co-transfection with this virus and this virus for getting the high number of virions being produced, which all will be having your gene of interest. Um, then the problem is that if you do transfection with both of these, then it was found that it is comparatively difficult to remove the helper virus. When you're harvesting your protein of interest, there is a chance that, let's say, your virus component or virus can also get purified, etc. So in that context, it's not that good virus. So every virus has its plus and a minus point. And just for your introduction, yes, these are the 4 different viruses which are most commonly used. So what, let's say if you are working on a retrovirus, I'll just briefly tell you that how will you proceed in the lab for cloning or making a stable cell line, or even for a transient transfection, etc. So let's say this is your target cell, some epithelial or mammalian cell line in which you want to get your gene transferred. Let's say it's not very well transfectable with your lipofectamine, etc. So in those cases, what you will do, that you will take a retroviral vector, like PBAB vector is one of the retroviral vector. You will clone your gene of interest here. Then you will infect this retroviral vector through lipofectamine in a highly transfectable cell line, which is HEC293 cell line. It is very highly transfectable. So with lipofectamine, so you won't have any problem here. So you you transfect it in here. So now these cells have your gene of interest, or let's say your plasmid has gone inside. Now, because your plasmid vector doesn't have the packaging things with it, so you have 2 options. One option that is also means 2 options, I would say, in various labs, they are equally used. One is that you co-transfect your gene of interest, your plasmid, with 2 other plasmids, both of which make the proteins which are involved in the packaging. So one leads to some transducing protein expression, and some leads to expression of the envelope protein of the virus. So what will happen, that now your 3 things are there, your gene of interest and the packaging systems are there. So now what will happen once this gets infected, so it will in 24 to 72 hours, it will start producing viruses, and each of these viruses will have your gene of interest. So now what I have to do is that I have to just harvest my virus. How will I harvest my virus? I just have to collect the supernatant after transfection from my mammalian cells. So once I harvest the supernatant, which has all the virions, and each virion has my gene of interest, I will infect this in the final cell line in which I want to make a stable cell line. So I will infect with, means I will make dilutions of it, and to ensure that only virus comes and no other particle from the bacterial origin comes and nothing else comes, I pass this supernatant through a filter, and then I dilute it, and then I infect my target cell line. And then if I incubate it, what will happen? My retrovirus will infect it, and then it will deliver the gene of interest inside the cell, and then that will make cDNA, because it will be RNAs, it will make cDNA. cDNA will get incorporated into the human cells or the cell line that I'm interested in. Then each of these cells I have to separate separately. I mean, I have to dilute in single wells. So, and then from each clone, I will get a multiple, colony being formed. And then I have to screen each clone, that which of these clones is giving me the highest expression of the vector. Why do I have to screen for my recombinant clones? Is due to the fact that the insertion is random in nature. So because of that, I have to find the recombinant clone which is giving the highest expression of my gene of interest. So then once I get that stable clone, I don't have to do repeated transfections or infections. Now I have, let's say, my CHO cell line in which my gene of interest is permanently incorporated in its genome. And what I have to simply do is I have to grow it in a bioreactor, okay, to make my protein of interest in huge amount. One thing that I just forgot is that I told you that one strategy of transfer of my vector into the cells is one. I told you that you have to co-transfect it with the packaging vector, right? But one option is also that now 293 cell lines are available in which the packaging these genes which are important for the whole assembly of the virus, they are already cloned in its genome. So these are called as 293 producer cell lines. And as a result, I just have to transfect my expression vector here, and because my cell line is already producing the envelope protein and the transducing the other proteins for its assembly, so I can the virus will anyway be made. So these are the two methods of transfer of your vector into the mammalian cells, and this is the whole process. First, you have to make a virus which has your gene of interest. Then you infect that virus to any target cell. It could be CHO cell or the Vero cells, which the various cells that I just talked about. And then you can use it for commercial purposes or for your lab research purposes. So the pros and cons of virus-based methods. Of course, you are dealing with virus. So you have to be very, means take a lot of precautions. You have to work in BSL2 facility for biosafety level 2 for majority of these viruses. And because these are viruses after all. And I think that you might be now remembering the COVID virus and its spread, etc. But I would like to say that majority of these viral vectors, they are very weak in nature, and but still you have to work in containment environment. Many viruses can also be lytic in nature, so you have to take care of that. Means you have to use those viruses which don't cause much lysis of the target cell line. You need spatial cell lines in where the packaging constructs are already cloned there. Then this method is obviously much more labor intensive. So in my lab, for example, I'm a mammalian cell culture lab. I would definitely go for lipofectamine based method, because for at least cancer cell lines, lipofectamine works very good. At least 50 to 80% of transfection we see in the cell lines that we work on. But let's say if my lab starts working on primary cell lines, then lipofectamine based method is definitely not good. And then maybe I'll go for these lentiviral or retroviral based delivery methods. But the advantages are definitely there. Like I told you, for lipofectamine, it is in the range of 50 to 80%, 90%, depending on which cell line we are using. For 293, definitely it is extremely good, like more than 90% we see transfection. But for other cell lines, even in my lab, we see like 70 to 80%, not reaching 90%, etc. So for virus, definitely the transfection efficiency is extremely good. And also, once your virus is made, then the infection is very simple, and you don't have to waste money. See, the method that I am dependent on, which is lipofectamine based, it requires repeated investment of the reagent. Means every time I have to buy lipofectamine and then do the transfection in my cell lines. But in virus, once your virus is made, you just have to incubate your cell line with the virus supernatant. That's it. And then the gene will be transferred. So pros and cons are there. Then there are so many reviews in which means so many success stories, in which you will find that so many proteins have been made, which are being
Excuse me,
used.
ma'am.
Yeah, I'm just done. If that's the thing, this is my. Yeah, I'm just done. So there are many success stories in which you will find the expression of various therapeutic proteins or enzymes which are lines, and you can read those reviews. And just a brief introduction to the biopharmaceutical market, that currently majority of the proteins which are used for therapeutic purposes, they are made in mammalian cells, even though it is much much expensive as compared to E. coli and yeast. So this is the general distribution that is currently seen. And for the enzymes, I see that at least in this particular pie chart, mainly it's yeast and followed by bacteria, where these recombinant enzymes are made. There was one recent article in which they say that there is a possibility that over time we go for cell-free expression of the recombinant protein, in which cell lysates are used rather than these growing cells for protein production. So maybe in future those will be the major things that will be used for recombinant protein production. Okay, so that's all from my side. Any questions?
Ma'am, the coming Friday, do we have a class?
If